So it's my pleasure to introduce Robert Skelton, or Rob Skelton. And I know him from a while back. He was a postdoc in Tasmania with Tim Brodrib. And then he was in Berkeley, I think it was right with Todd Dawson, on another postdoc. And this is the study that we're about to see. And currently, he's a researcher in the South African Environmental Observation Network, which is in Cape Town. So he presents us data about safety margins in oak, different oak species. And Rob, really thank you for agreeing to do this. And, stage is yours.
Thank you, Uri. Let me share my screen. Okay, now I guess I get to see what it's like talking to a fairly blank screen. Interesting. Yeah, thanks Uri for the invite. It is very disconcerting not having a single face to talk to, but I'm gonna just assume that everyone will appreciate my laughs and will laugh hysterically. So to get into what I'm going to talk to you today, as you'll notice, the title of my talk is Evolutionary Relationships between Drought-Related Traits and Climate Shape These Large Hydraulic Safety Margins in North American Oaks. And you might be wondering what a South African is doing talking about oaks, but so a little bit of my research. How do I get to the next slide? Okay. Will hopefully make some of that clear. So this is where Uri and I overlap. We're both plant physiologists. Uri has investigated grapevine and a few other species. I come from some fairly diverse systems, so I enjoy thinking about diversity of plant physiology. So I describe myself as a comparative plant physiologist. And so what I seek to do is to generally understand plant form and function, and to explore some of the evolutionary and ecological patterns that drive this diversity of form and function. And so testing hypotheses in this field is fairly complicated. What it requires is identifying functional traits, developing mechanistic or conceptual frameworks of how plants respond to abiotic stress, and in particular, I research drought tolerance. And then it involves testing these hypotheses using natural and lab-based experiments. And so today I'll briefly touch on a few of those natural and lab-based experiments that I've been conducting over the last few years. And hopefully the outcome of this research is firstly an improved understanding of plant form and function. There are a lot of advances that we need to make in this realm. And can you all hear me, by the way? Is this? I'm not getting any response, but okay, I'll assume that it's all working well. And then secondly, so once we've identified these functional traits and we understand them a little bit better, okay, I've got some thumbs up, that's good. We can take quantitative metrics from these functional traits and use this to predict how plant communities, how species are likely to respond to future drought events. So again, if you're wondering what a South African is doing measuring oaks and talking about oaks, I started off in the Cape Floristic region where I've been researching Mediterranean type species in the wonderful fynbos, the wonderfully diverse fynbos. I then did a first postdoc in Tim Brodrick's lab in Tasmania. Again, there's just an amazing diversity of plant species in this part of the world. And then for a second postdoc, I moved to California with my wife, and we got interested in the oak diversity of California. So really, my background is thinking about diversity and thinking about how plants can tolerate these relatively dry or semi-arid environments. So that's a little bit of background to me. This talk, I've tried to break it up into three main components. The first two I have slides for, and then the third component I thought might direct some of the discussions that we might have after the talk. So the first bit is I'll explore some of the conceptual ideas of how we think about plant physiological response to water stress. So I'll discuss the prevailing paradigm of plant drought tolerance. And then the second bit is where I'll explore some of the results from our recent postdoc in California, thinking about or attempting to quantify the physical tolerance limits of North American oaks. So for those of you who saw Uri's talk about 2 weeks ago now, there'll be a little bit of overlap in the first part of this talk. As you will be able to tell, plant physiologists like to get onto the same page when it comes to understanding physiological principles. And a lot of the principles that Uri communicated also apply to a lot of the plants that I've been working on. So how do we start to think about a plant and how it functions? How do we start to think about plant form and function? We do this by one of the ways we can do this is to think of a plant as analogous to an electrical circuit, where you have a bunch of resistors, and then so you have water flow through the plant, and you can identify gradients of water potential. I'll spend a little bit of time discussing water potential because this is often a concept that a lot of people find fairly difficult to grasp at first. So it's the idea that plants don't have a heart. So how does water move to the tops of trees? How does water move to the tops of plants? And the idea is that there's a continuous stream of water connecting the soil and the roots through the xylem into the mesophyll of the leaves. As the sun hits the mesophyll, that energy drives the evaporation of water molecules from the evaporating sites within the mesophyll tissue, and so water. This creates a tension, a negative pressure in the water column, and as a result, water is drawn up through a plant. So you can think of it like a rubber band. As a water molecule evaporates because of evaporative the energy from the sun, that water molecule that essentially pulls the water column up through the plant. So another way to think about it is in terms of water potential. So we can think of the potential of water to move. So the metrics are fairly difficult to comprehend at first. We talk about negative pressure. So this is, just think of this as that tension. The larger the numbers, the greater the tension. But it's important to remember that water potential, as it applies to plants, and negative pressure starts at 0 and then becomes more negative. So water moves from areas of high water potential, in other words, water potential close to 0, to a water potential that's fairly low, that's more negative. So one of the physiological responses that plants have evolved to limit water loss as conditions get drier is to close their stomata. So we can think of this as a variable where as conditions become drier, so as the water potential decreases, it becomes more negative. Plants can limit the evaporative water loss from the leaves. And we have an image of a stomata that's closing and reopening and closing, and that's to prevent excessive water loss. It's one of the physiological mechanisms that plants have evolved to limit desiccation or to restrict desiccation. So the next idea that we've hit upon as physiologists is the idea that the xylem, the water column in the xylem is breakable. Plants can have what are called plants have heart attacks, so this is called embolism. And so what we can see is that as the conditions in the plant dries out, these resistors within the plant xylem also become variable resistors. And we've developed techniques and tools to identify this. So again, as Uri showed with those really neat images in grapevine, we can examine embolism as it occurs in a leaf. So this is a series of images on the left here of a plant that's drying down, and so there are about 1,000 images that are being looped at the moment. And what you will see eventually is that at a particular point, this image on the right shows the image difference between two successive images, and what you see is the plants embolize, the leaves embolize, and this is, as you can see, it occurs in the veins, the xylem of the plant. So this is essentially where the rubber band snaps, and these processes, what's happening here is that air is either being drawn into the xylem conduits, or air that already existed as air bubbles within the sap has expanded, and then these air bubbles expand and form blockages in the xylem. So these are what we're referring to as plant heart attacks, and we can do the same thing for stems. Again, a series of repeat images of a dehydrating stem. You can see some early events in this plant, and then as the video progresses, as the plant dries down, as that tension increases, and as water potential decreases, we see more and more events in the stem. So we can track these processes as I mentioned, in leaves and stems. And this is a relatively recent technique. It's only been developed in the last 5 years or so. Okay, so what results from all this understanding of the kind of basic physiological processes that occurred in response to water deficit is a paradigm or a conceptual framework of how we think plants respond to water deficit. And this is captured in this diagram, in this illustration here. And what you can see is, what we expect rather is that there's a phase of recoverable decline of function. So this is indicated by the green phase here. Plant physiologists are, again, because we think in terms of negative numbers, we think backwards. So we think from right to left, so what we can see. So this is essentially the sequence of responses that we would expect, moving from a hydrated state to a dehydrated state. And what we can see is that there are turgor-driven processes of function within the plant. So plants open and close their stomata. This is recoverable. loss of gas exchange capacity, loss of function. Plants can shed their leaves. This is a deciduous oak from California. I have my finger on the new bud, and you can see the current year's. So this is next year's leaves, and the current year leaves are senescing, and then we enter into a phase, as you can tell from this blue line here, where the plants experience embolism. So this is the damaging or non-recoverable process that I indicated in the previous two slides. And then we enter into a phase where the plants lose function in the distal twigs. There's dieback within the branches, but the plant is not yet dead. As you can see, the plants have this remarkable capacity to tolerate severe water deficits. So they, as long as there's some meristem in the plant that's still alive, they can resprout epicomically, for example, in this oak, and they can recover. But with each of these progressive stages, there's a lower recovery of function. And then finally, ultimately, the plants incur too much embolism, too much damage, and they fail to recover. So this is kind of the typical sequence of physiological responses that we have identified. And what we can do is we can take some important metrics to try to describe these processes. So the one is we can define the turgor loss point, so the point at which the plants lose turgor. We can define P50, indicated by this purple dot here. So P50, which is a trait that I'll talk about quite a bit. P50 is simply the water potential associated with 50% loss of hydraulic function, or 50% embolism, as evidenced by this curve here. And this, just in case I slip into the vernacular quite a bit, this is what I refer to as a xylem vulnerability to embolism curve, or a vulnerability curve. And again, I'll be referring to P50, which is one of the metrics that we can take from those curves quite a bit. So what we've seen in the literature over the past couple of decades is our attempts to try and integrate these functional traits, these mechanistic traits, P50 and the like, into predictive frameworks based on functional traits of how plants are likely to respond. One of the kind of major conceptual advances was done by Brendan Choate and a host of other authors, where they compared minimum water potential. So this is the seasonal minimum water potential. So during the driest part of the year, what water potential do plants typically get down to? So the larger the number, or the lower the number, the more negative the number, the greater the tension and the more stress the plant is under. And what the paradigm that's emerged from this type of thinking is the idea that plants have converged on the safety margin. And this is the safety margin that I was referring to in the title. This is called the hydraulic safety margin. But what you'll notice is that there are a lot of species, particularly from Mediterranean woodlands, that have negative hydraulic safety margins. So these are the species that occupy this portion of the curve. So these are surprising species where they seem to tolerate, or they seem to experience regular loss of hydraulic function in any given season. And there's a non-trivial number of species in this group. It's approximately 22% of all species experience greater than 50% loss in any given season. And we can, if we zoom in on some of the oaks, we can see that this pattern is is pervasive amongst oaks. So this is I have extracted data from the literature this is a lot of this data comes from that choke data set, which was a meta-analysis. And what we can see is that a lot of the oaks are predicted to lose a lot of function during a regular season. So some species have P50 values that are very high, that they're very vulnerable. The P50 values are close to zero, and we can see that these species are unusually or surprisingly that they close their stomata prior to this P50 value. So the question is, what's going on in this instance? And just to take one species to explain how different this is from the prevailing paradigm or the prevailing framework that I outlined earlier. If we have an expected curve for this one species, so this is Quercus gambellii in the deserts of Arizona and Utah in western United States. So if the blue line shows what we maybe expect just based on our fundamental principles of physiology, with knowledge of the Turger loss point and where the species loses complete function or the lethal water potential. And yet the published curves for the species are all highly vulnerable. So this is one of the species that has been considered in a number of papers in the literature where the P50 values have been shown to be quite high. So it's quite an unusual. There's something unusual about either about the physiology or about the metrics that we're using that doesn't quite fit the paradigm, the prevailing paradigm as we now know it. Okay, so one of the potential issues is that there are some authors have indicated that there are methodological artifacts associated with quantifying these vulnerability curves. And so one possibility is that the metrics that we're seeing are artifacts of dealing with negative pressure. So if you've ever tried to deal with a rubber band, they're quite tricky to deal with. They always want to they want to snap back, and when we deal with cut plants, when we deal with water in the xylem, can lead to some fairly unusual results. And so what has emerged is that the two prevailing paradigms when it comes to or rather two hypotheses when it comes to plant function. So these have variously been referred to as the rare embolism or the embolism avoidance hypothesis versus the common embolism or the damage and refilling hypothesis. And so it's important to consider these two things. The first is that there are these methodological artifacts, so we need to find innovative ways to try and figure out what the true vulnerability curve is. And the second thing is that each of these two hypotheses produce quantitative predictions that we can test. And this is where I started researching the North American oaks. So we brought out a paper in 2018 where we collected cut branches from the field, and we had some potted individuals, and we showed that these species, that some of these species are not as vulnerable as they have been suggested to be in the literature. There was a bit of pushback against this, which is one of the ideas is that we've missed early events in the stem. So with the optical technique, you have no way of assessing whether there were early events that that you might have missed. So we've done some follow-up experiments where we've gone to the Lawrence Berkeley lab in California, in Berkeley, and we've scanned some of these potted individuals. So these individuals that have no damage, that haven't been cut, and what we see is that early on there are in this case there are no events, or there are very, very few events. So we know that we're capturing a plant that hasn't been seriously or hasn't been compromised in any way prior to scanning. And then we follow these plants as they dehydrate, and we show that at a particular point, embolism starts to increase quite rapidly. So these data support the results of the optical technique quite nicely. And this paper we're currently working up at the moment. But again, my interest is more in diversity. I'm interested in exploring the variation of these physiological these physical tolerance metrics in these different species, and this is where I started researching all of the oaks of California and a couple of the oaks of Western North America. And really the research was driven by this question: so what is the interspecific range and capacity to withstand embolism in Quercus? I wanted to explore this question in more detail, and then the other two questions are dealing with very specific predictions from this embolism avoidance hypothesis. The one is that there are these quantitative thresholds are likely to be aligned with the climatic niche of the species, in accordance with the rare embolism hypothesis rather than the common embolism hypothesis. And then another prediction of the rare embolism or the embolism avoidance hypothesis is that these plants should have fairly large safety margins, which prevent embolism in any given season. So I'll discuss some of these results from this research. And so what we did was we measured the physical tolerance limits in about 20 of these oak species from 4 different sections of oaks. We had the red oaks, the intermediate oaks or the golden cup oaks, and then the white oaks. I think there are 2 oaks in Israel at least. Maybe there are a few more, and I think they're from the Ceres section. So we don't have any of the Ceres, but I've worked with Sylvain Delzon and Laurent Lamarck, and on some of the so Quercus ilex and Quercus robur from the same section, and the results seem to agree quite nicely with this with the results that I'm about to outline here. So we had a diversity of oaks. These oaks occupy, as I alluded to earlier, very, very different environments within North America. So they occur in temperate rainforest up in the Pacific Northwest near Oregon, and then the environment becomes drier as you move further south until eventually you get into the deserts of the Southwest. These are some very, very dry deserts. Some of them are high elevation deserts, as you can see in this image here. This is Bryce Canyon in Arizona, I think. And again, so you have these very different species occupying these different environments. In terms of the phylogenetics, variation in physiology can be understood better if considered in light of phylogeny. What we have in North America is these three main clades, which are sections in this instance, have entered independently into Western North America and then radiated from that point. So we have these three separate lineages, and two of the lineages, the red oaks and the white oaks, have also entered into eastern part of North America. So Uri might be familiar with some of the white oaks and the red oaks from his time at Harvard. But the oaks of California within those sections form monophyletic clades, so we can assess patterns amongst these closely related species. Okay, so here are the results. What we find is that there's approximately two to threefold variation in P50 of these different oaks from these different vegetation types, these different plant communities. Plants of the temperate rainforest in the Pacific Northwest tend to be more vulnerable in terms of P50, so they tend to have P50 that's closer to 0, indicating that they're more vulnerable than species that occur in Mediterranean chaparral, Mediterranean type scrub, or desert scrub, as indicated by the species here. And then the oaks of the Mediterranean woodlands in California tend to be intermediate in terms of their P50. And so what's important to note, well, one thing that's important to note is that these results suggest that even plants of these fairly mesic environments have high capacity to withstand air entry. So again, this pattern is very different to the pattern that you saw earlier from the literature. So again, we find support for some very different patterns emerging. So when we place this variation in a phylogenetic context, we see that species within each of these clades or each of these sections are very similar. So closely related species have very similar P50 values. In the Lombardy, so these are the red oaks. There's hardly any variation amongst species. This is similar in the Protobalanus or the Golden Cup oaks. And when we get into the Quercus or the white oak, the white oaks, what we see is that there's more variation within this clade. So some species, Quercus garryana, for example, are relatively vulnerable. They have a P50 of negative 3.8 or so, and then a couple of these species in this smaller clade here, the scrub oak clade, some of these species can be quite resistant. So the toughest species we have is Quercus pacifica, which occurs on the Channel Islands, just off the coast, just off the mainland of California. The species is quite resistant to embolism, and we can assess some of these patterns through time. There has been a trend within this part of the world to become cooler and drier through the Miocene in particular, and it's interesting to speculate as to whether this might be driving some of the patterns that we see here. But I don't want to spend too long on that particular question. So the next question we can try to address is whether these physical tolerance thresholds agree with the climate, the climatic niche of these species. And what we see is that the climate predicts quite nicely the variation in P50. And again, this pattern is somewhat different to what we've seen based on some of the data that are in the literature. where there are a lot of very vulnerable species occurring in some very dry environments. And again, we think that this might be a result of artifacts associated with these techniques. So when we use the optical measure, we see a fairly linear relationship between climate and vulnerability. So predictably, species that are occurring in dry environments have more resistant xylem. So the final question that we can ask is whether this physical capacity to withstand embolism forms part of a predictable strategy or set of behavior of different species, incorporating a number of other functional traits pertinent to water loss, and whether this results in embolism avoidance in the field when combined with the site that the individuals are growing in. And what we see when we combine our P50 measure, our metric of the capacity of the xylem to withstand air entry, when we combine it with some of these other morphological traits, we start to see that there are clear patterns of association between the xylem traits and some of the other traits. So here we have leaf length, leaves, which is a proxy for leaf size, with the idea that larger leaves they might have a capacity to lose more water, whereas smaller leaves might aid in water conservation. And what we see is that indeed smaller leaves are associated with species that have greater capacity to withstand air entry. And we see that the species of that can tolerate water deficit also tend to be smaller in stature. And this can be that information can be summarized by this cartoon, where in general the taller trees that we see that tend to be the more vulnerable individuals with larger leaves, whereas individuals that have greater capacity to withstand air entry in both leaves and stems tend to be lower in stature. They tend to have smaller leaves. So there appears to be an architecture associated with drought tolerance. And it'd be interesting to think about, is it Quercus coccifera, I think, is the one that occurs in Israel, whether that's a low, the prediction would be that that's a fairly low growing shrub with fairly small leaves, and as a result it might be predicted to have relatively high capacity to withstand air entry. So when we put all this data together, when we assess the water potential, the minimum water potential of these plants in the field, what we with the new optical data, what we see is the predictable patterns that we predicted from the conceptual framework of plant drought tolerance appear to be consistent with the optical vulnerability data. So minimum water potential is likely to be a result of variation in aridity of the growing environment. It's likely to be a product of an integrated product of plant architecture and other plant water use traits. And again, what we see is that these species tease out very differently when we compare the optical data with the previously published data. So these species seem to be adhering to the paradigm that has emerged. And again, if we look at the turbo loss data, this is the stomatal a proxy for stomatal closure. What we see is that these species tend to close their stomata after they've experienced, before they've experienced 50% loss of hydraulic conductance. And again, these data are consistent with this embolism avoidance hypothesis, the idea that embolism is this very late phase event that plants are trying to avoid in their environment. And when we look at the hydraulic safety margins, again, what we see is in some species there are fairly large safety margins, and these species tend to be the individuals that occur in fairly dry environments, although this pattern is not perfect. You do have some species that occur in fairly mesic environments that have fairly large safety margins, and then you have some species that are functioning on the verge. So there are a few species from the Mediterranean woodlands that are functioning very close to this threshold of catastrophic hydraulic damage. And the idea is that these species are the species that are most likely to be impacted by future drought events, by future global change. So these species on this end here would be species that are functioning relatively close to the edge, whereas these species have a fairly large safety margin, so they can experience maybe greater or more severe droughts and still have some margin from experiencing catastrophic damage. And when we assess the stomata safety margin, so this is now the difference between when the stomata closes and when the plants lose 50% hydraulic conductance, we see that most species tend to close their stomata well before they experience this catastrophic loss of hydraulic conductance. Okay, so I think we're coming fairly close to time when I to the end of the presentation, at least. I wanted to leave some time for discussion, but just to conclude, to have 5 main takeaways from this presentation. What we've shown is that some of the surprising patterns that are presented in the literature for Mediterranean woody long-vesseled species, those surprising patterns disappear when we use new techniques that potentially avoid some of these methodological artifacts. And what we show is instead is that even species occurring in fairly mesic environments have xylem that is capable of tolerating fairly substantial water stress. And what we find when we look at a phylogenetic pattern is that there are fairly large differences between clades, but that closely related species within each clade vary little in capacity to withstand air entry. And we can speculate as to whether this allows us to better predict the likely physical tolerance of other species that we haven't yet quantified. Whether we can use the phylogenetic context to maybe predict some of the variation in these traits that we might expect to observe, because it's very difficult to measure these traits and to generate some way of better predicting the function of diverse species is essential. And then what we find is that the growing environment of these species influences their hydraulic traits. Again, potentially unsurprising, but what we find, which is different to some of the patterns that have been shown in some of the other oak communities, the Floridian oak communities, which have intermittent freezing and waterlogging, is that the climatic niche strongly predicts the dehydration tolerance, and that closely related species within these Western US communities tend to occupy fairly similar similar niches. And then finally, some of the evolutionary relationships between drought tolerance traits. So this is things like leaf size and plant height, combined with these climatic this climatic variation tends to yield minimum water potential values that avoid catastrophic embolism within these species. And this line of inquiry demonstrates that these oak species are not, or some of these oak species are not yet on the verge, and there is some buffer to experience lower water potentials. All right, so with that, I'd like to thank the research hosts for this work, in particular David Ackley, shown here in the optical cave, the dark room where we did most of these experiments. Things tended to get quite busy in those rooms during these campaigns, and then Todd Dawson as well, and Sally Thompson, who was these were the three kind of supervisors on this project, and then a lot of people that I've worked with, many collaborators, Leander Egg, Jess Diaz, who did a lot of the assessment of these optical images. It's exhausting, time-consuming work that we're hoping to automate in the future to make it a little bit easier. And then some of the other individuals that contributed made theoretical advances to the study. A lot of lab group assistants and students who helped on the project, and then I was fortunate enough to be able to travel the width of the breadth and width of California, because there's a fantastic natural reserve system within the state that promotes or accommodates researchers quite well. And then finally, my recent time at the South African Environmental Observation Network has been funded by the Royal Society and the Global Challenges to Research Fund, and that's allowed me the time to investigate some of these patterns and processes. And lastly, thank you everyone who I assume has either fallen asleep or is wildly entertained. It's hard to tell. I'm happy to take some questions.
Thanks, Rob. Anybody want to ask a question? Who's first? You have to unmute yourself, by the way, if you want to ask a question.
If I may. Can you hear me?
Yeah, we hear you, Shachar.
Yes?
Rob, I have a question. How does it relate to, have you ever looked into fruits? I mean, what would be the P50 of fruits rather than leaves?
Yeah, interesting. So so fruits, yeah, they do have xylem, and there are a couple of people that are measuring sap flow into roots. I haven't come across too many people that have measured embolism of pedioles of fruits.
Not roots, fruits with an F.
Yeah.
Fruit.
Yeah.
Not the root.
Yes. Yes.
All right.
That's it, correct. So again, I haven't. What I've seen is people have measured sap flow into fruits, but as far as I'm aware, no one's measured embolism within fruits. I have seen some images. Someone showed me some micro CT images of what we assumed were embolism events within apples, but I don't know in terms of the physical capacity of those fruits. I would just be speculating. A few people have looked at flower variation in floral capacity to withstand air entry, and flowers seem to be a lot more vulnerable than some of the distal tissues of those individuals. And so that's research that's coming out from Tim Brodripp's lab, and there's a group at Yale, Adam Roddy and Craig Broderson have looked at floral venation and embolism patterns within flowers. But in terms of fruits, yeah, it would be interesting to explore that further. I think this paradigm, if I can go back to some of my slides, this paradigm. How do I share my screen again? This paradigm that I showed here, I think there are a lot of this paradigm that I showed here, there are a lot of processes in this range here. Leaf senescence, leaf shedding, fruit production, and how those processes, dynamic processes within those in those responses. I think those are fascinating questions, but I don't yet have the answer for that.
Thank you.
Anyone else?
I have a question.
Go ahead, Jonathan.
Thank you. There is a difference between a deciduous and evergreen oak.
So this, how do I answer this? When we've compared the two, it does appear that some of the deciduous species have greater segmentation between leaves and stems. So segmentation is the idea that leaves embolize prior to stems. But the pattern, when you include all these species, that difference tends to disappear. So we know that there's at least one species that we investigated quite a lot, Quercus douglasii, which is the blue oak, which is actually that species that I showed with the leaves that are senescing. We know that the leaves are more vulnerable in that species than the distal twigs. And in fact, there's a paper that came out this week looking at some of the hydraulic architecture, some of the anatomy. This is the group that you've also worked with, Uri, Steve Jansen, that kind of group, where they've looked at some of these architectural, some of these anatomical conditions within the pediole, and they do find that there's greater hydraulic resistance within the pediole, which might influence these processes. So yeah, I think it's a great question. I don't think it's as simple as saying that deciduous species would have more vulnerable leaves than evergreen species, but I certainly think there are species that are worth investigating.
Okay, thank you.
I have a follow-up question to Jonathan's. So do you think that in those cases where the safety margin was 0, meaning that probably the leaves can cavitate, so in most of your oaks, did they really cavitate, or were they shed before these P50s were achieved? like when you were going through the forest, everything was green, and you think that the leaves contain 50% loss of conductivity, or it's just because you dried them in the lab?
Okay, I'll answer that by showing this picture here, which is, so this is Valley Oak, this is Quercus lobata, which I think does embolize. Now this was a fairly severe drought within California. So this was that 2014. This is when I first arrived in California. There was a severe drought, and at this time, which is where a lot of those water potentials were taken, you do see embolism within these. Well, you see evidence of embolism within these trees. Now, so your question might have two another component to it, which is, I know you've shown that there's variation in vulnerability within a season, but I have we've done a lot of work where we've quantified we've quantified patterns. We've tried to quantify patterns within the species, and although there is so these are 7 different populations from along California, and in this one species. So this is that blue oak species. So we have these 7 populations in a common garden and in the wild, and we see that there is variation within these populations, but that when we compare the populations, that it doesn't seem to be any regard. Plasticity, once the xylem is fully mature, I think it is reasonable to say that this is the P50 of the species. So I think the xylem matures, and maybe it's a little bit more vulnerable early on in the season. But then once you have secondary xylem that's laid down, and the leaves are fully mature, I think the values are comparable across populations. And so I think the idea of whether we can measure P50 and whether it's meaningful, I think it is meaningful. So the next question is, do I think that they're experiencing 50% embolism? In some instances, I would say yes. These individuals have often shed their leaves, but there is still cuticular water loss or residual water loss after that point. So I think their minimum water potential does still decline. Yeah, but it'd be really interesting to put these little clamps out there and to try and quantify that, to try and capture that. But I would think that yes, we are seeing embolism within the stems as well as the leaves, in these fairly dry seasons.
Thanks.
I don't know if that
is
your question.
May I comment regarding the leaves? In our Mediterranean climates, drought is most often associated with heat. And when the leaves close the stomata, the first thing you see is the leaves are burned. They're suffering heat damage rather than drought damage, I mean. And so you see the branches becoming bare due to leaf burning rather than hydrological or hydraulic processes.
Yeah, and I wonder if those two things are coupled, right? So either the plant's not losing any moisture because the stomata is so tightly closed, that might be true. But in addition, you might have embolism in some of those peripheral vein networks that then prevents any movement of water to those distal sections. So I wonder if it's a yeah, if you know, it'd be interesting to know whether you have dieback in the absence of embolism, or whether you always have embolism that then results in dieback. Yeah, the leaf scorch patterns are very interesting. There's one person who's investigated patterns in avocado, and they found that embolism was involved in the scorching in the scorching events within those individuals. But yeah, it's really I think there's so many interesting questions around kind of interactions between these traits, and that's kind of where I was trying to go with the section on leaf size and plant architecture. I think it's the trick is trying to think like a plant. They have they're very different to animals, and but I think there's sense to be found in combining all those traits and trying to develop some kind of holistic understanding.
Okay. Rob, thank you very much. I think we should all start thinking like a plant more. And take this take-home message.
If you take one thing from today, that's it.
So thank you very much for this talk. It was great.
Thanks, everyone. Yeah, it was quite an experience talking into the internet, but anyway, it's fun. Thanks for having me.
Bye bye, everybody.